Okay, okay, thank you. Welcome, everyone. So, I'm just here to introduce Fernando. So, Fernando Barroso is a professor of biomolecular science at the School of Pharmaceutical Sciences in the University of São Paulo in the Ribeirão Preto campus. He has a PhD in theoretical chemistry from Lund University, and his expertise is in molecular biophysics, and particularly in protein electrostatics, also on structural bioinformatics and computational virology. So Fernando's group develops computational techniques for biomolecular systems, and also more recently studying antibody and antigen recognition using the techniques he's developed for biomolecular interactions. I've also, known Fernando for quite a while, and we've been collaborating on various projects. And he also has been a researcher and visiting professor in Sweden and in France and in Ireland. So I'll just, today he will be talking about the molecular aspects of Zika and COVID virus. So I'm going to just let him.
Thanks, Eric, for the kind introduction. Thanks to the university for hosting me here. It's really a great opportunity to talk with you all, either in person or online. I think the plan for today is more or less to translate things that we do in the basic science to more, let's say, in the infectious diseases problems. And I will try to highlight or to flash you with some ideas, some problems that we have been solving. And let me just start to make a few comments about how it's the plan and talking about what, in general, we do in our lab. So the goal for today is to start with this very brief introduction about what we do. Then next, we are coming to one of the, let's say, initial problems that we have attacked in this field, that's Zika virus, which happens to be very important in Brazil, in particular after 2015, where the number of microencephalic cases have increased a lot. And next, I jump to SARS-CoV-2, which I hope you were not really tired of to hear about it or to suffer a lot from this problem. But I think the SARS-CoV-2 was a nice experience in terms of science, at least, where we could do in simulations a little bit different than often we do. For the first time, we could predict things before we had experimental data. So we could use the experiments that came later to validate the simulations and not the opposite way that is often the case in simulations. So this was, challenge as a human health problem, but it turns out to be a very challenging and nice opportunity for science to advance. So let's start with, let's say the goals of our lab, the general ideas. What we do is to develop and apply constant pH simulation methods, and in particular, we are interested in biomolecular interactions. So could it be the interaction between a protein and a nanoparticle, protein polyelectrolyte, protein-protein interactions, protein RNA. And in this case, we are trying all the time to explore the physical chemical environment like pH and salt and mutations. To do so, what we work on it is to developing these codes, so writing our softwares as well, using, of course, available tools, and applying these to interesting problems. And one of the interesting problems for us, in particular nowadays, is virus and the binaries, both for therapeutics, but also for prevention diagnosis. So this is, let's say, the general framework, what we are doing in our lab. The general idea is to take a very complex system, and I'm starting here showing you one example that's not directly related to virus, but just to show that once we dominate these biomolecular interactions, it turns out to be appealing to translate it to different problems. So let me start with one problem that was probably the topic of one of my previous talks here at Tennessee State, where we work with food proteins. So in this case, we try to make, for instance, microencapsulation. And in this problem, we try to identify what are the key elements. For instance, in this example, we have a polyelectrolyte and a protein. And we make a toy model where we can study these things in a computer and get results. Example of measures that we can get. Well, we can get titration plots, as you can see here, charges as a function of pH. We also have free energy of interactions, so free energy as a function of separation distance, virial coefficients, and so on. Now, I can change completely my problem to something much more complex. That's the biological system, where for infectious diseases, we end up looking to something specific, like we have here our antigen and our antibody. So again, we are back to two proteins that are going to interact with each other, or two molecules, macromolecules that are going to interact with each other. And we need to pick up the key elements, make a model, and use this toy model for predictions. In this case, since we are playing with pH, this happens to be very useful, in particular for pH-responsive problems. An example, we have the pH-responsive antibodies. So this is also a hot subject. For instance, when we are working with the cancer, I mean, we haven't done any work yet with cancer, but cancer is an example that you need to work with lower pH conditions. So it's important to have in the model the pH for the system, also for practical reasons. Another feature that's very important, that these simulations, they need to be fast. I mean, molecular dynamics simulations are really very useful, but if you need to repeat your simulations in many different scenarios, changing mutations, changing pH, and changing salt, we needed to try to find simplifications in order to make it useful, and to, at least as an initial screening of the process, that later you can specify a couple of cases to run more detailed simulations. So we try to work with these constraints. And again, we can come up with the same kind of measurements. So I change the problem, but we get essentially the same quantities, titration plots, free energy of interactions to measure the binding affinities. And in the case of the antibodies and virus in general, we can come up measuring the epitopes, and both at the surface and the sequence level. So we have developed a couple of models, I mean, trying to balance between the number of sites, I mean, the CPU costs, their accuracy, and their practical relevance. So this is typically things that we try to balance. So depending on the application, we put more details and we pay a higher price. Depending on the applications, we can decrease the level of resolution of the model. So we try to play with these things. I'm not going to all the details, but the whole idea is that we can define what kind of the approach, what kind of spherical call, if you come from physics, is going to be like a very typical example, what kind of level of resolution we need for that particular question. So depending on the problem, you select the appropriate tools. So this is, let's say, a very general framework. Just to flash you some examples of tools that we have been developing. I mean, we started with colloidal-like models, and now we are able to run titration schemes together with molecular dynamics using this, what we call the combination of FPTS, our fast proton titration scheme, with OPEP6, that is a force field for proteins, a coarse-grained force field for proteins. So, and also we can combine these things in an approach that is going to be very much useful for the applications that I'm going to talk about. That's called the 40, the fast coarse-grained protein model, where we basically have the two proteins that can translate, they can rotate in any directions, and this is going to be a function of pH and the environment conditions. So in this kind of system, running a Monte Carlo simulation, we can sample the probability to have the system in different separation distance that can be transformed in free energy of interactions, and can be used also to get the amino acids that are more important for this process. So this is, let's say, the core of most of the models. Okay, as I said, let me jump in the details just to show applications to you. We have been using this kind of models to weigh proteins, food proteins. We have been using this to biomaterials like spider rings. We have been using it to septins, and also in drug delivery systems. So these are examples of different fields where these simulation tools can be used. And over the years, maybe during the last 10 years, we have decided to attack one very important system, virus. Well, if you'd like to simplify things, we can say that when you can start with the capsid of a virus, that's essentially is a protein-protein interaction system. But this protein-protein interaction system is a little bit more special. If you compare protein-protein interfaces in general, you can see that they have one kind of a pattern. But when you look at the protein-protein interface specific for antigen and antibodies, they are much richer in titrable groups. So we have much more amino acids that can titrate, which means that this is much more appealing for electrostatic problems and more appealing for pH simulations, because in this case, we probably have a richer electrostatic features of the system. So all the experience that we had before with electrostatic problems can help in this field. In fact, now starting moving to some examples, let's take a look in Zika. Zika, for most of the people, is going to give you minor symptoms. So typically we don't have anything that serious. But it turns out that, especially when we had the crisis in 2015 in Brazil, people started to have microencephaly. So babies were born with this terrible problem. And this is something that everybody was curious. I mean, what happened with the virus for this reason? So this was a motivation of part of our work. I mean, first of all, to understand why the virus in Uganda was not giving microencephaly and why in Brazil it was giving microencephaly. And also to think about what kind of diagnosis tools can be useful, because PCR is typically very expensive, and especially in poor countries, this can be a problem because the diagnosis is typically based on clinical symptoms. So it's not that efficient if you really would like to be precise what's going on, to just use a description of your symptoms. Well, now another, let's say, problem that we are facing is dengue. I mean, this has been around for quite a while, but this year in Brazil, it has exploded the number of cases. I'm sorry for the Portuguese slides, but I think they are much more appealing just to show you what's happening. I mean, between 1 week and another week in March, I mean, this data is from March, you can see the large increase in the number of dead people. I mean, we had 363 in 8th of March, and then it jumps to 561 dead people in just the very beginning of the year. And you can see that the numbers in 2024 are already bigger than all the previous years. And I mean, what we have observed in 4 or 5 months of the problem of this dengue crisis this year is much larger than what we have seen in all the year of the 2015, 2019, and so on. So we really have a very big problem. And this is not just a problem for, let's say, tropical countries. You can go, for instance, to New York, and you're going to have this message at the airport warning you to protect yourself against Zika. So this is a picture that was taken in LaGuardia. And so it tells you that the problem can be not just located in a tropical place, but the mosquitoes are getting adapted. There are other mosquitoes that can eventually transmit the dengue and Zika. So it's better for us to start spending more time on understanding their molecular basis to see how we can better not just improve ourselves, but to improve the whole community and our neighbor places. So I'll jump very quickly here to the flavivirus genome in general. You can see that there are several different proteins that were involved in the genome of this flavivirus. I'm going to focus here for one special reason in the NS2 protein. And NS2 is one of the non-specific proteins of this virus, and it's involved in several parts of the life cycle of the virus. And for this reason, it's very appealing for diagnosis. And our hypothesis that if you compare, for instance, Zika from Uganda to Zika from Brazil, we have some reason why, in the case of Uganda, people were going to be dead soon, while in Brazil they can develop microcephalia related to NS1 protein. So our hypothesis here is that the affinity of the virus in the NS1 dimer with the membrane is much higher for the Ugandan strain than it is for the Brazilian strain. So this is what we have worked to demonstrate by computer simulations that can be a possible explanation why people are developing microencephaly in Brazil and not in Uganda. The simple reason is because it was a faster end of the person. I mean, it was much more aggressive, the virus in killing the person. So there was no time to develop microencephaly. Okay, now let's try to connect the two things. I mean, I said about electrostatic features, and then I said about the virus. I said specifically about NS1, and you can see several experimental papers. I'm just pinpoint here one example where we have a clear difference between the electrostatic features of proteins, NS1 proteins from different flavivirus. In this case, we are comparing here dengue 1, dengue 2, West Nile virus, and Zika virus. And you can see the difference between the blue and the red plots all over the surface, indicating they have really a very diverse electrostatic potentials. So when we saw this data, so comparing, for instance, dengue 1 and Zika, we said, well, what about the difference between Zika from Brazil, Zika from Uganda, and from different strains? So what we have done is to look to their phylogenetic tree and identify some key elements or select some of these strains, some of the sequence from these strains, and use these two lineages of the virus to explore their features. In this case, you can see here an example of a plot of the charges as a function of pH for strains from Uganda, from Senegal, from Brazil, and a couple of others, as you can see here. And clearly have very different patterns for the electrostatic features. So if they have different charges, if I select any pH condition here, you can see that the difference can be quite large. It's obvious that they are going to have different interactions with their partners. So the protein-protein association, the different oligometallic states of the NS1 as a dimer, as a tetramer, or as an hexamer is going to be different. Also, the interaction of the membrane, as our hypothesis, is going to be different depending on the net charge. Plus, of course, the interaction of the antibodies or any kind of therapeutics should be affected. So this is, let's say, a summary of let's say, the general ideas or things that we could identify just looking to the charge as a function of pH. So you can draw these kind of conclusions. Well, in order to measure what amino acids are really playing a role, it's the time that I go back to the basic physical chemistry. And then I use something that I have done together with Eric a while ago to establish how we can identify key amino acids. And these ideas were evolved to define what we call here the PROCEED-PKA method. The whole plan is to map pKa shifts due to the presence of other charged molecules. So in other words, I'm using a very basic physical chemistry knowledge to identify the preferential bind spots of the molecule. And doing so, we can map which amino acids are going to be critical, that sometimes they happen to be not just at the interface, but they can be a little bit deeper inside the molecule. And if you titrate or you affect their protonation states, this can also be propagated. This is something that we have seen before for protein polyelectrolyte interactions. So in this case, just to remember you about this the basic physics, if I have here a tetramer groups and I have a neutral charge, they are going to remain, for instance, in this case, suppose that their pKa is around 12. Now, if I come to the same molecule and I place a positive charge, the pKa shifts are going, the chemical equilibrium is going to be shifted. So in this case, I can get a little bit smaller number. So if I'm able to monitor in which part of the protein, which groups that were affected by the presence of the other molecule, I can directly identify the ones that are more critical for the binding, for the interaction of the two partners. So this is the idea that we have done before, mapping, as I said, protein polyelectrolytes and protein interactions. So in this case, we could map for a couple of proteins these hot spots. And it's the same idea that has evolved to develop the PROCEED-PKA. Of course, we have it compared with other traditional tools. And you can see in this rock plot here that we are doing relatively well, in particular for flavivirus, where we have benchmarked it. Okay, using such things, you can take the basic physical chemical data from a simulation and use analytical tools to predict the dimer association. You can predict the dimer membrane interactions in a very, let's say, crude and preliminary, but fast way. So you can just not go into all the details, but you can see that the plots are different. And if the interaction is different, it indicates that there's something going on. That's the obvious thing that you can see from these plots. Also, the way that we can see the interaction with antibodies, as I already mentioned to you. So this is, or let's say, the very, early data that we had for this problem. Later, we start doing a little bit more sophisticated things. So we start running molecular dynamics, classical molecular dynamics simulations, in this case, not using our own tools, but using GROMACS and available force fields, both, as you can see here, at the atomic level, and some versions of coarse grain methods. When we compare the two, let's say, NS1 from Brazil and from Uganda, we can already start seeing some differences, like I'm highlight here some numbers that appear to be different. Like you can see that their surface accessibility is getting different. So it indicates that they have some peculiarities that not just in the charge level, but also how these charges are going to help the molecule to organize itself. So we can see this also in terms of the coarse grain model, in this case, look to the rates of duration. So the features were there. If you compare the root mean square fluctuation to have a feeling how is the structure of the proteins and the dynamics of it in different contexts, we can already see that there is a tendency for one particular region, some amino acids at NS1 from Uganda to be a little bit more flexible than they are in the Brazilian strain. We could see it in the streams as well, and it is middle position around 110, 120. If you do a PCA analysis, you can clearly see that they are exploring different spaces. So you have here the plot for Uganda, and here the plot for Brazil, and they are really definitely they are exploring different day space. We can also confirm this data in the coarse grain modeling. So the coarse grain model is captured to this difference, although they are not equal, but they kept the same nature. Well, we have done some dynamic cross-correlation metrics. I mean, we have done a couple of analyses, and I think the take-home message here is that even a small difference at the sequence level can give different interactions with their biological partners. This is probably the main conclusion that we can come at without going into all the details. Now, there was another hypothesis then that could raise. I mean, what about their association process? Typically, you need the NS1 to be at least in the dimer to start the process. Often you need an hexamer. So can we predict in this model how is the association of the virus, I mean, these NS1 particles, from the monomers to become a dimer, and from dimer to go to the tetramers, and so on. And another thing is, if we can do it, how is going to be the interactions with the nanobodies or antibodies? So let's start looking to the interactions with the antibodies. In this case, we can see a pH effect. So you can see that depending on the pH, like here in blue, pH 3 is essential repulsion. Here in pH 11, we are back to some similar situation. And when you go to pH around 5, we have a very strong attraction. So this is what you can observe for one of the NS1, in this case from West Nile, and one of the available antibodies. We have about 3 to 4 antibodies that are known in the literature. Well, we can map the regions, we can make the comparison. You can see the difference between West Nile and the 2 strains of Zika. And here you can identify the key amino acids. And in each case, sometimes you have the same amino acid that appears to be important in all the 3 sequences here, others that are important in a given one. So this helps you to understand what the difference in the sequence is given in terms of the estimated antigenic regions. Remember our hypothesis, so in terms of the affinity with the membrane, so we have performed analysis, and we can identify that there is really a difference between the way much more, let's say, attraction that we can observe for the Uganda and Brazil measured by different criteria. In this case, I'm just highlighting you here the number of amino acids involved, interactions with this PO4 for the Uganda and for the Brazilian. You can see that you have a large number of amino acids that are interacting with PO4 than you have for NS1 from Brazil. Furthermore, I mean, if you look at the energies, you can also see that in terms of the NS1 from Uganda, you have an attraction for most of the properties that indicates or suggests at least that this interaction is more favorable. So there are, let's say, an accumulation of evidence that NS1 from Uganda strongly bind to the membrane, and bind is so strong that it can destroy the membrane and so on. You help to kill the patient. Although in Brazil, this interaction is weaker, and being weaker, eventually you give rise to the microencephaly. So this was, let's say, one of the conclusions that you can draw from these studies. Moving on, I mean, in terms of self-association and NS1, you can see that the number of experimental data is quite limited. For instance, we had for dimers some electrophoresis data only for dengue, one of the strains of dengue. So you can see that in different pH conditions, they exist as a dimer or as a monomer. Oh, sorry, monomer was dimer. So we tried to see if the computer models could predict it. So it was, I mean, we needed to predict if you can get a stable complex in the proper pH condition. But that's all the data that we could find for experiments, and it was specific for dengue too. So there was no experimental available information for Zika or the other strains of of dengue. So we performed simulations to try to see if you can find dimers, tetramers, and so on. For instance, here you can have the data from NS1 Uganda, Brazil, dengue 2, and West Nile. You can see that in PH3, there is not really a dimer formation. There is no tetramers, so no association. You can see that the presence of sugar or lipids are not necessary because we can start forming dimers in pH7. So this was another hypothesis, how sugars, glycans, or lipids were really important for the self-association. In a simulation, you can remove these things and test if they are really important or not. I mean, so you can see that my free energies here are getting negative and indicating that they can form a dimer at this given condition in a model where sugar and lipids were absent. And the binding is going to be increased if you go from dimers to tetramers. As you can see, the numbers here are showing a more negatively free energy of interaction. Okay, I think it's good, I mean, to give you this an overview. I'm jumping now to SARS-CoV-2. In the case of SARS-CoV-2, at the very early days of the pandemic, what we have done was to convert everything that we were doing for flavivirus to start to work with this virus. So when we saw this paper in January 2020, it was the moment that we have started to work on it. We got the sequence of the virus. There were no experimental data at that time. We only had the sequence that was published. And using the sequence, we had hypothesized, which we all know now, that was the receptor bind domain that would interact with ACE2 receptor. So there was at that moment in January 2020, one PDB structure with the receptor bind domain from what we call now SARS-CoV-1 interacting with the cell receptor ACE2. So this was the experimental available information. Here I'm just highlight again the receptor bind domain in the spike protein. We know how this is important for all the pathogenesis of the disease, all these things, but let's focus on the simulation for a while. So at that moment, I mean, since there were no experimental data, we could have this opportunity, as I mentioned at the very beginning of my talk, to challenge the models, I mean, to bet in our predictions. And what we did was to take the sequence that was available. I mean, we had the sequence and the structure of the SARS-CoV-1 receptor bind domain. So we will use this guy here, which has a 76% of identity. So we use this guy to build up a model of the receptor bind domain from SARS-CoV-2. If I compare what we have done in that time, this was before AlphaFold. So what we have done here using homological modeling is that our receptor bind domain has an RMSD around 0.6 to the structures that were solved later. A couple of weeks later, we had an avalanche of new structures that came out. And even if I compare structures among themselves, they have RMSDs around the same range. So the predicted model was quite reliable, especially if you compare using these metrics. And we use this model for our biomolecular interactions. So we hypothesize that the receptor bind domain, you'd use the cell receptor ACE2 as SARS-CoV-1, and we'd like to demonstrate that it could be possible. So we have performed our protein-protein interaction simulations, and we could in fact see that this was the case. So the binding affinity for the receptor bind domain happens more or less the same separation distance, and it should because the particles are about the same, and they are relative equivalent to each other. Using PROCEED-pKa, this method that goes back to the basic physical chemistry, we could map the number of amino acids that were involved in both viruses. And in this case, you can see that if you go for SARS-CoV-2, we have more amino acids that are involved in these interactions, which turns out to be in agreement with experimental data that was published after this work was done. So it was also nice to see that not just the binding affinity were, the binding affinity measurements were relatively well, but also the number of involved amino acids, although it won't have a necessary, the same comparison, the same quantitative comparison, they qualitatively agree with experimental data. In terms of the binding affinities, the data is quite scattered. I mean, you can see labs that have measured different labs. So I think we can safely say that they are about the same range. Well, we have also performed pH studies to explore the virus in different pH conditions. So we can see, for instance, going from pH 4 to 7, there is an increase in the binding affinity. But I'm not going to have time to show you these things. Let's go back to the idea that we had that time. I mean, in terms of now talking about more treatment. So on those days, very early days of the pandemic, there were only 4 known antibodies to bind the receptor binding from SARS-CoV-1. So the plan here was, okay, let's try to see if one of them, I mean, could bind to the receptor binding from SARS-CoV-2. And if they could block this interaction. So this was our question mark here. So we took it in and performed the same kind of simulation, protein-protein studies, but in this case, using the structures of the virus, or in the case of CR3022, for instance, the very early days, there were no structure available. So we had to repeat the same trick with homological modeling to get a model from the sequence. And doing so, we could see that this guy was the best candidate. So this guy was the best candidate, but you can see that the binding affinity was a little bit less than we can have it for SARS-CoV-1. So it was less efficient. There were papers later confirmed that SARS-CoV-2 receptor binding could bind to this antibody. So it could confirm again the experimental results. There were also crystallography data that could show the structures of this one, and there are many more that came later. But we tried to say, okay, can we improve this model? Can we improve this antibody for something better, at least theoretically better? So we have come up with a pipeline here where we take the structures, we use our tools, and we measure in a kind of loop way all the main features like the binding affinity, the epitopes, and the possibility for these antibodies to find aggregation, and also if they are stable. So in this loop, we do a kind of alanine scan. So we replace one by one the amino acids, and we repeat the cycle again. That's an advantage, again, to have something faster than other tools, because this allows us to explore in a systematic way the contribution of every single mutation. We have done this cycle up to 3 mutations, 3 simultaneous mutations. When we try to go to 4 mutations, we end up having stability problems of the final mutated version of the protein. So we stopped in 3. And doing so, this scanning procedure, we got a little bit better improvement of the molecule. You can see that now the numbers between SARS-CoV-1 and SARS-CoV-2 are much similar. So we can see that playing around with the mutations, in particular for the electrostatic amino acids, these are the 3 amino acids that were mutated, we could improve the binding affinity. Okay, and also the number of involved amino acids were a little bit increased in this case. So back to the history of the life, I mean, we start having all this soup of letters, alpha, beta, gamma, and so on. I mean, so that time, one of the main constraints was if the up and down states of the molecule could help the transmission. So we could address this question, trying to predict, I mean, how would be the electrostatic stability of our proteins in different conformations. I mean, you can see here the up and down movements. Remember that the receptor binding domain has to be in the spike protein has to be in the up state or open state so the receptor binding domain is going to be able to bind to the AC2 receptor. And we checked if we could predict this stability. So it turns out that if you compare SARS-CoV-1 and SARS-CoV-2, in this case, we started with the wild types, we can see that the stability is very much different between SARS-CoV-2 and SARS-CoV-1. In SARS-CoV-1, it's much more stable for all the 3 states of this protein. So here I have down, down, down. So it means that all the 3 spike chains are in the down position. When you see a letter U, it means that one is up. So you can have this combination, and you can see that in general, it's much more stable for SARS-CoV-1. When we try to make like an estimation, we can see that for SARS-CoV-1, it's more or less 66% of the time the virus has or the probability for the virus to be in the up state is 66 more or less. When we are in SARS-CoV-2, it's more or less 50-50. This helps in some way to understand why the transmission of the virus by asymptomatic people has increased, because eventually there are more people with the three spike proteins in the down states. So something could trigger them to go in the upper position. And if it goes to the upper position, they eventually evolve to symptoms. That's just a hypothesis that I'm going to show some data that helps in some way to support it when you compare to clinical data. Let me now just make a comparison again for SARS-CoV-2, for instance, from the wild type and the beta variant. In the case of the beta variant, we can see that there is a higher chance for the virus to be on the up state. So you can see that it's way much more stable on the up state. So over evolution, the virus, some of the variants at least, they got much more stable on the proper states that should be ready to invade our cells. Okay. So, by the way, this is also what happened with BA3, one of the Omicron variants that appeared and very rapidly disappeared. So the virus already ready to enter cell. If I try to summarize, I mean, of course, we have been testing different variants here. So just to summarize, if you see what I'm saying here is like SARS-CoV-2, the wild type, has a kind of chameleon feature to be more or less in the same probability to be in both states. The beta version, the one that appeared in South Africa, is more probable or all the time probable to be in the up state. So if you look at the clinical data, I just took one data from this group here, where they show that people that were contaminated with the beta variant were all the time symptomatic. So probably because they have all the time the spike protein in the up states. Well, this is, of course, there's a big gap between the clinical data and what we are doing in terms of modeling, but there is something that we can go closer, is if you look at your cryo-EM data. And cryo-EM data, for instance, for this variant shows that more often it's true to be in the up states, as you can see here in this data. Well, another feature that we have explored is polymorphisms. I mean, genetic variants that everybody can have. So different populations have a tendency, and we can see that some people that were born with some genetic on one of these polymorphisms can have a higher chance to favor the interaction of the receptor bind domain and ACE2. So typically people with Italian descendants can have this mutation. So it's very common in European genetics. So it favors the attraction between the receptor bind domain and ACE2. Another very nice thing that you can do with electrostatic models was to benchmark, I mean, to get pKa analysis and try to correlate them in this kind of network diagrams. In this case, I'm showing you what we can get from pKa. So we have performed pKa calculations from different variants and different mutations. And you can see that they cluster in a particular way. Like I have here the gamma variants, beta. I have here the Omicron family. And you can see that they are organized delta in a kind of independent group. And if you look at the phylogenetic tree, they happen, I'm just jumping here some results. Oops, sorry, my plot just has disappeared. But if you look at your phylogenetic tree, we're going to end up seeing exactly the same thing. So you can reproduce phylogenetic trees just using electrostatic features. I'm sorry about that. I mean, there was supposed to be a new slide here where you can see a very similar plot with phylogenetic tree. Anyway, another feature that we have been exploring was, can we predict the evolution of the virus? I mean, how far we can go, where we are. So what we have done was to use a couple of mutations. So we select from the known variants up to 2021. We have been selected a couple of them and measure their bind affinities and their charge. So we can see that as the time of the pandemic is going over, it's increasing their charge and also increase their bind affinity. So the virus is getting more efficient to be attracted by our cells, which is not good news for us. Just to point to you, I mean, in this plot, it's not updated, but we have done this for the variants that appeared after the publication of this paper. And we can see, for instance, that JN1 ends up here. Remember that BA.3 is the one that is an exception because it tends to be all the time in the down states. So in a model that we are just focused on the receptor bind domain, not simulating the whole spike, this movement is absent, but at least we can see the same trend. I mean, we can see that it's more efficient. It's not going to be that efficient in the long range because we are missing part of the information. But if you, for all the other variants, we end up in a very nice prediction. There are, of course, much more things that we can do in such models. I mean, this is something more beautiful. Say, oh, by the way, here I already have the data for a variant that was circulated more or less 1 year ago also, XBB 1.5. It's another Omicron variant. That's probably the one that you were taking as, I mean, you're using to produce the last version of the vaccines. So it fits in this plot. But I was saying that the other nice thing is that this is a very nice example where biological science and physics, they work together. I mean, from the analysis, I mean, pure analysis of the data, just use linear regression of this plot, a very basic machine learning approach, if you'd like to say. You can extract this plot, I mean, this line, and this equation happens to be very similar to what we often use in colloidal science as the video theory. So you can get the physics just using the experimental data, which happens to be something quite nice. Okay, we have tested a couple of antibodies that were available, more than 28 different antibodies for different variants, so we can map them. And we identify again that CR3022 could be improved. I mean, it was one attempt. I mean, there were others that could also improve. But we decided to stick to the first one that we have done, and we proposed a new cycle. I mean, what we'd like to show here is that one tool can be useful, but if you combine more than two, you can gain more. So what we did was to start with Rosetta. We feed up Rosetta with the experimental structure that became available. We select our top 20 candidates from a very big set that was produced by Rosetta. Our main criterion was to have Rosetta unit energy that were more attractive than the original protein that we know now because we have the experimental data, and also that the interface area should be equivalent. So these were the thresholds that we have used to select our top 10 candidates. We also have visualized the structures to make sure that there was no steric repulsion or anything wrong with the side chains or like a crash here in the interface region between the RBD and the antibodies. And doing so, we took the best top candidates. We submitted them to two different approaches. One was a coarse-grained simulation with the CETA force field to get the free energy analysis. So it was done in a metadynamics study. At the same time, we have performed atomistic simulations to double check the results from part of the coarse-grained simulations. We have also used our own approach, I mean, the Forte approach to get the free energy. This is the cheapest one. So we can explore many more scenarios, different scenarios here than we can with CETA force field. And we can compare them to see, first of all, if there's a consensus, but also to identify if there's anything that one method is better to capture than the other. So doing so, we could also take the results from Forte and repeat the electrostatic optimization that I already mentioned with the alanine scanning and getting the best antibodies that were, again, tested against the known variants to see if they work. So the main result, I mean, there are lots of data that we have done, but in this case, I'm just showing, let's say, what is more, let's say, relevant for this discussion, is we took the top 10 candidates, we compared the results here from the different approaches. So you can see here that there was a consensus for one candidate. This is the one that we have nominated our lead candidate, where we had all the consensus. You can see the structure here with the receptor bind domain. The results in green are the ones from Rosetta. The results in red are the ones from the constant charge simulation. I mean, pH just defined at the very beginning of the simulation, and then the charge remained constant. And we have used CETA from umbrella sampling. And then we have our own approach, Forte, where it's a real constant pH simulation, and we have discharge fluctuation mechanism present. You can see that the black and the red plots, they follow more or less each other, although there is a display here, but they follow the trends. One possible thing that is good to highlight that since we can run longer simulations with Forte because it's cheaper, we can get smaller estimated errors, which better allows us to classify the difference between the amino acids, the candidates. And we can see here that for Forte, we also got P6 as a possible candidate. We decided to keep the analysis using P1. It's slightly better than the original wild type. As you can see here, the affinity has decreased. And this was being measured by all the tools. And we tested this antibody with all the possible variants from that time. I mean, when I say Omicron here, I mean BA1. We can see that our individual, let's say, if you use just our own tool, we got this result here in red. If we start using Rosetta, we can see a big improvement. But if we combine Rosetta with this pipeline where we have the electrostatic optimization as well, we can improve even further. And a nice result was that although we have been focused just on the wild type, it turns out that it also works. I mean, the optimization was done only for wild type, but it turns out to also work for Omicron. Another feature that we can see is that how electrostatic is important. I hope now that we can, I mean, we are running some experiments with some collaborations to try this in vitro at least, these antibodies. So they are synthesized and they're going to measure their binding affinities. So we can have a better conclusion if it's working, or if it's not, if you can gain more information to verify or to rethink about how the hypothesis that we have in our model that eventually can improve then for better antibody engineering. But I would like also to, again, highlight here all the features that you can see from the electrostatics for all the models that were generated. So there is a promising way to design better binders. Just to highlight some things before you were too tired of me, I hope that I could show you that it's possible to use very simplified computational models to address very complex problems in biological science, in particular in infectious diseases. So electrostatics is really playing an important role. I'm going to make a comment about it later, but they can help you to design new applications. I think this antibody engineer is a good example. In the case of Zika, we can see that the virus evolution has helped the virus to be less attractive for the cell, although it kept its, let's say, capacity to form dimers and so on. So it can keep, I mean, the viral production capacity. Zika is one example that's more related to this case, I mean, about the interaction with the cell. In SARS-CoV-2, we can highlight that it's going a very strong evolutionary process that really is enhancing the electrostatic properties. And our predictions indicate that the virus has still room to be more attractive by the cell. So this is the bad news for us as a human being. So there is a chance to increase the transmission, unfortunately, unless we think about some measurements. Maybe we should all be wearing masks, by the way. What else? I think that's it. And with that, I would like, of course, to thank you for your time. Thank you for the time and the people that were watching us through Zoom. And there were lots of people that were involved. I mean, all the time we have a work as a team work. I will start with the students that were directly involved in this project, really specific for Zika and for SARS-CoV-2. So I have here Sergio that was from the Zika project and Carolina Giron that was for SARS-CoV-2. There were people that were very important to develop the basis of most of these methods, including Eric Santizo. So thank you, Eric. And Deepti was the student that was involved in this project. And all the partners that I have with Caterina Tibast, Atulaxo, Andrei, and Francesca Mossi that were more involved in the SARS-CoV-2 platform for antibody engineer as well. So we had to run several calculations, and a big effort was important to be able to repeat this, to do many replicas and these kind of things. And of course, I would like to thank the foundations and the people, the taxpayers that support us through these different organizations. And again, thank you for your attention. Now, of course, I'm happy to take any questions. So, Michelle.
Yeah, I'm just gonna get closer.
Okay.
So people can hear me, but you can still stand there because you're gonna answer. Okay. The first question is, what is the reason for such a big spike in dengue mortality? Is it localized or spread across Brazil?
Well, if I go back to the plots from the distribution in terms of Brazil, you can see that it's spreading all over the country. I think we have a couple of reasons that we can see. Let me just get the plot. Whoops. That's a problem with animation. It takes longer to go back. And I think, of course, one reason is that we have this change in the weather. So it's getting warmer and the mosquitoes are multiplying faster. So more mosquitoes, faster multiplication. So more people getting contaminated. And this is one thing. But it seems also that the new variants are also more efficient in the interactions. So that's another feature that might be the case to explore. If you were asking for the distribution, like, come on, where's the plot? Okay, jump it too quickly. Sorry, guys. And I'm afraid to change here, otherwise here you have the distribution. I mean, you can see that when you have these red states here is where you have more than 500 cases per 100 inhabitants. So it's more concentrated so far, more on the southeast, but we have here in the north also some states. And in general, I mean, this is just a matter of time probably to go all over the place. I think it's going to be related to the rain season. I mean, we are more, let's say, having the rain season on the south. I mean, it's raining a lot, much more than it used to be, by the way. And this in some way helps the mosquitoes to be multiplying.
But it's funny because you would expect that in the north where it's warmer to see more infections, right?
Yeah, but I think
less developed.
There's another problem that we were discussing. I mean, we have to be careful with this data because, as I mentioned, the diagnosis is typically based on clinical symptoms. So if you go to a doctor and you have symptoms, we don't know how reliable it is. I mean, in the case of the SARS-CoV-2 and the virus that are transmitted by air, we are trying to convince some towns to use the waste system as a measurement to see what kind of...
they do this,
yeah, so that's something that we are trying to convince people to use it. But for a virus like Zika, to be better to use the blood. And that's a little bit more tricky. I mean, we try to talk to people from these Hemo centers to see if they can do the analysis. So this can be an estimation about the number of real cases, because just based by clinical diagnosis, I think there are people that eventually are contaminated by Zika, for instance, and they got the information that they were contaminated by dengue, or chikungunya, or any other of these. Yeah. So that's a problem. I mean, to reply, this is the data, but I don't know how reliable it is. I mean, we have to put some question marks. For sure, the number of cases is exploding because you can see that even in your daily experience that you have people that are all the time complaining. And actually, I was joking, but it's true. I have a PhD student that happened to arrive in Brazil, and he's working with dengue nanobodies, and nanobodies against dengue, and he got contaminated in his first month in Brazil. So yeah, he was not careful enough to use the creams.
Before I moved to the States, I had dengue as well. And, they scare you a lot because if you get it reinfected,
yeah,
there's a much higher chance that you get the hemorrhagic fever, which is not fun.
Yeah. So this is to answer the question, I guess.
Yeah, and then there's another question in the chat. I'm not sure how to pronounce it. If it's Guillain-Barré syndrome, is typically associated with specific Campylobacter strains. I believe you showed the cases in relation to Zika virus infection. Do we know if these patients also were infected with Campylobacter? I don't know.
I don't know, but I'm sorry, I don't know.
Okay. There is another question. It says, in your last slide, you said that you were experimentally testing the antibodies to see if they were working. Are you mainly getting binding constants, or are you trying to see if they're block binding to ACE?
Okay, for us, what's really, really important at this stage is really to get the binding measurements. So we would like to see if in real life, the binding affinities, they are at least in a relative scale equivalent to what we are getting in the simulation. So this is, let's say, our main goal. But the experimental people that are supposed at least to perform these experiments, they are the ones that are really looking for neutralization. It's a biopharmaceutical lab, and they would really like to make sure that they can come out with a new antibody in the market. So we have different aims. I mean, in our case, from the theoretical side, it's good to have some, let's say, feedback, how they are working, so we can know if the models are really suitable for these things, what kind of improvements we need to do in the models to make them more appealing.
That's all I have in the chat right now.
Okay.
I've got a question too.
Yes.
So I'm an entomologist by training and studying vector-borne diseases in mosquitoes. And one of the things that makes is most concerning for vector-borne disease spread is if a virus can like jump which species it uses as a vector or change its mode of transition. So like Zika was super interesting, not just because of the microcephaly effects, but also because it was then able to be sexually transmitted by humans, which is weird for
or a blood transfusion as well.
Well, yes, but the sexual transmission was to me one of the weirder things for a vector-borne disease. So I'm wondering if this kind of like protein-protein interaction and the models that you use are able to maybe predict which viruses are most likely to switch vectors or switch transition mode and make them more likely to spread broadly.
Well, this is a very good question and also a very good project, I would say. Because I mean, in principle, the models that I mean, we are talking about mutations in the virus, but we can also talk about mutations in the ACE2 receptor or try different receptors. I mean, when I showed you the data about the ACE2 polymorphism, in some way we addressed part of it because we are focused on humans. But in principle, we can repeat the same analysis, trying different receptors from different animals to predict the ones that are let's say, more probable, at least to interact with that particular protein from the virus. So in principle, this is something doable. I mean, it's like to ask the old dog to do the same trick again. It's more or less what we have to do. I mean, it's essentially, instead of just focusing on the mutations for humans or for the virus, is to try different cell receptors, or in some case, it's the same. It's just the same receptor, but with different mutations as well. So doable. Might be good. I mean, if you have suggestions, we can talk.
Mm-hmm.
There's something to check as well, right? I mean, now instead of further is jumping from one thing to the other.
Yeah. Well, in fact, I mean, SARS-CoV-2 is doing it. I mean, that's why it's going to be very, let's say, my opinion, that's going to be impossible to get rid of it. Because at some point, I mean, you can have a kind of box somewhere that can give you back the virus in a different version that can again start the process again. Oh, sorry. Sorry, guys, I was outside the camera. Sorry.
Well, so do we want to close the Zoom?
Yeah, well, I had a question, but I don't know. I mean, I can always ask you later. There was a point where you were showing a PCA plot comparing the Uganda and Brazil strains.
Yeah, I think we can find it again. Yeah. Just take some.
I thought that was really interesting because you had a very clear separation.
Yeah, they are totally different. I mean, you can see that you can see. I mean, you can see both for the atom simulations and for the coarse grain level.
But now, because this is PCA, in principle, you can look at the eigenvectors and backtrack what are the features that are contributing the most to each one of the eigenvectors, right? That's basically what you have there.
Yeah. We try, I mean, to summarize the things. I mean, it's, how should I say, at the very beginning, I mean, we're trying just to explore which direction should go, to trying to understand what were the features that could contribute for this big difference between what's happening in Uganda and what happened in Uganda and what was happening in Brazil. So we try to explore different features. Of course, now it's easy to tell the story, and I can simply find just one slide. We can see clear that was the binding affinity with the membrane. I mean, this is really what comes as, let's say, the strongest evidence what's going on. But we have signals that the two proteins, they were behaving a different way, despite they are very similar level at the sequence. So they are very similar in terms of sequencing, and they behave in a very different way.
It's funny that it affects the brain of the fetus and not the person who is infected.
Yeah, I think it might be related to the composition of the membranes. And again, what we have observed is for a model of a membrane. So we try to mimic a membrane from a human. I mean, what should be the composition of this membrane? I don't remember by heart, but we tried really to get how should be this composition. But eventually, this is not really the one that is going to be working for everybody, or for adults or babies. But at least it's clear that it increases its binding affinity. So.
All right. Well, yeah, we don't have anything else on the Zoom. Okay. So thank you.
Thank you for your time.
Yeah.